From the JAMA Network, this is JAMA Clinical Reviews, interviews and ideas about innovations in medicine, science, and clinical practice.
Hello, and welcome to this JAMA Clinical Reviews podcast. I'm your host, Dr. Revital, JAMA Fishbein Fellow and neurologist. Today, I will be speaking with Dr. Dan Roden and Dr. Josh Denny. Dr. Roden is a professor of medicine, pharmacology, and biomedical informatics at Vanderbilt University Medical Center, where he is also senior vice president for personalized medicine. Dr. Denny is the chief executive officer of the National Institutes of Health's All of Us research program and formerly a professor of biomedical informatics and medicine at Vanderbilt University Medical Center. We will be discussing the article that Dr. Roden and Dr. Denny co-authored, together with their colleague Lisa Bastarash, from the recent JAMA Insights paper entitled Phenome-Wide Association Studies, which was published in the January 4th, 2022 issue. Dr. Roden and Dr. Denny, thank you for joining us on the podcast today. Dr. Roden, welcome.
Thank you so much.
And Dr. Denny, welcome to you as well.
Thank you so much. Pleasure to be here.
Well, I thought we'd get started with some definitions for our listeners. Can you please describe what genome-wide association studies and phenome-wide association studies are, otherwise known as GWAS and PheWAS, respectively?
I'll define GWAS because that's pretty generic, and I'll let Josh define PheWAS because this really was largely his brainchild. So the idea of a genome-wide association study is to ask the question, at what genomic locus or loci are there signals that associate with a disease? So let me just take an example. You study type 2 diabetes and you find cases of type 2 diabetes and you find individuals who are otherwise similar but who do not have type 2 diabetes. We know that there are common sites in the genome where there are variants that are defined. So we can use a so-called chip-based assay to assay 500,000 or a million or several million of these common SNP sites and ask the question, is there a reference allele or is there a variant allele at that particular site? So then we do 1 million tests of association across the entire genome. And at every site where there's a known variant, we ask the question, does that variant associate with having type 2 diabetes or not? So we generate a set of 1 million tests of association. Now there's complicated statistics around that, but using that kind of approach, you can find places in the genome, you can find specific genes, and occasionally you can find specific variants, but really this tells you where in the genome there are regions that associate with susceptibility to common disease. So in brief, a GWAS starts with a human trait, often a common disease or sometimes a variable response to a drug or a variable laboratory test, and then ask the question, where across 1 million SNP sites across the entire genome is there an association with that particular trait? So a PheWAS turns that experiment on its head, and I'll let Josh describe that a little bit more.
Thanks, Dan. That's exactly right. And that a PheWAS is basically, in a way, the inverse of a GWAS. So in a phenome-wide association study, or PheWAS, instead of starting with a disease and asking what genetic variants might be associated with that disease, we're starting with something like a genetic variant. So we may have taken that very genetic variant that Dan talked about in a GWAS for type 2 diabetes, and then we get to ask the question, what diseases might that genetic variant be associated in a phenome-wide manner? So when we look across a set of genetic variants, we're often maybe looking at 1 million or more different variations in the genome. The number of diseases we look at is much smaller. Typically in a PheWAS, we may be looking at 1,800 different diseases that have been aggregated typically from billing codes. There are many ways you can do this, but the way we've done is we've taken the ICD-9 codes, the ICD-10 codes, looking both across versions that happen in the US and internationally, and map those to a common architecture, a common nomenclature of diseases that we can ask what of those diseases are conditions that a given genetic variant might be associated with. And this really highlights when a genetic variant might be associated with multiple different kinds of conditions. So the type 2 diabetes study, perhaps a given genetic variant might also associate it with type 1 diabetes. It might also associate with gestational diabetes. These kinds of things you could see come out of a PheWAS. Sometimes you see things that are even different, very different. Maybe it's associated with a completely different kind of condition that you may never expected. And that is really one of the powers of looking phenome-wide across, for associations with a given genetic variant. And I'll just say one more thing is this is the kind of thing that was really only possible once we had electronic health records linked to genetic variation. Before that, you knew about the individuals in your study, the diseases that you asked them. And so there was always going to be bias by what kinds of conditions you asked about. But in an electronic health record, you're going to miss maybe detailed phenotypes you might not get some other way, but you really get a pretty good record of the things that would drive an individual to interact with the healthcare system. The diseases, conditions, signs, and symptoms that are going to drive them to interact with the healthcare system. And it's not going to be biased by, what a given study was looking for. And so that's one of the powers that's emergent from a PheWAS and electronic health records.
Thank you so much, Dan and Josh. That's very helpful. I appreciate you explaining that. I am curious how the PheWAS studies came about, how you became interested in them.
So in the mid-2000s, we were planning a biobank at Vanderbilt. And the rough outline was that we would have a biobank that would use the electronic health record as the phenotyping platform. And then we would get genetic information and link that to the phenotypes. And we started collecting samples in 2007. And we had this idea that we would start off by developing methods. Josh was going to develop methods to find cases and controls for common diseases. And then we would do a genotyping exercise where we would genotype probably 50 common variants that were known to be associated with these common diseases. So type 2 diabetes was one of the diseases that was studied. And by then there had been enough GWAS to know what variants were strongly associated with type 2 diabetes. So we genotyped something like 50 common variants that had known associations with common disease already. And then somebody had this idea that said, well, once we have the genotypes, we could ask the question the other way. We could say, well, here's a variant. With what human disease does that variant associate? And that was the first time I heard that.
My version of history is similar. I would working in the electronic health record to apply natural language processing algorithms to clinical notes that providers had written. And with this, I had been deep in the electronic health record. I had been amassing these data sets that allowed us to ask questions and help find these phenotypes that a given patient would have that would support the genome-wide association studies. And that included billing records, that included medications, clinical notes, all these kinds of things. And we had been looking at other ways of using billing codes and natural language processing to define ways of figuring out all the diseases someone would have. So as we thought about this, it's like we can do something with this data set. And just this phenomenal story of thinking of how a person's interaction with the healthcare system can tell you so much, and wanting to leverage that, learn the richness of that, and learn from the depth of an experience someone might have with the healthcare system, and not be biased maybe to the question you were asking initially with a genetic study, but really letting that full clinical experience shed light on your interpretation of a given genetic locus was some of the power of answering the question in that disease hypothesis-free approach.
Thanks for sharing that. That's very neat to hear how this all came about. Josh, you were explaining about PheWAS and the use of the electronic health records and billing codes, and that one really starts with genetic variation as a starting point. Can you give some examples of how one does this?
So the first thing we thought about is, what kind of variants should we pick to do? And then how do we actually do the PheWAS? And the variants, we picked a handful of variants that we knew had associations we were expecting to find. Because the first thing when you do a new kind of analysis like this is you want to actually be able to show that it works, that you demonstrate you're able to find what you're supposed to find. And of course, have the potential to learn something new that is plausible. So we picked a set of 5 variants that we knew had associations. And then we decided to run the test against ICD codes. At that time, we were looking at ICD-9, and there were about 18,000 ICD-9 codes, which is too many. You're not going to have statistical power. And many of these codes, anyone who practices medicine knows, there's not necessarily a lot of reason why an individual might get a specific type 2 diabetes code. There are certain complications there. There are certain things that might drive you to get one or the other, but they all represent type 2 diabetes. So there are many codes that represent type 2 diabetes that you want to group together. And so what we did is we went through and I built a big Excel spreadsheet and made groupings of these codes. Then I actually went through and did some chi-square analysis amongst all the different codes to help find new groupings that might be there, to pull together, for instance, all the tuberculosis codes, which were disseminated across a variety of spaces in the ICD-9 hierarchies. And so we built this nomenclature. At the time, it was much smaller, it was less than 1,000 codes. And we tested against these 5 variants. And we found that we were able to replicate a number of associations that were known. The strongest of these was an association known with an HLA variant and multiple sclerosis. And we also identified the possibility of some other associations which might be known. That was the first study. It was reassuring to us. And then we performed a much larger study across 5 sites in a network called the Electronic Medical Records and Genomics Network, eMERGE, which has 5 sites in it that all have genetic data linked to electronic health records. In this study of about 13,000 people, we were able to test a much broader range of hypotheses, replicate a bunch of different associations. Overall, 66% of those associations, which were adequately powered to detect a signal, and then discover new things. And we were able to validate some of those new associations as well. So we were able to really show that a given genetic variant could be associated with multiple conditions, some of which we didn't know before.
That's fascinating and very intensive how much work went into getting all of this set up. I didn't quite appreciate that. What happens if there's an error in the electronic health records and how things are coded? We know that sometimes happens as clinicians, especially when you're integrating data sets across multiple institutions. How do you account for that? What kind of effect does that have on this?
That's a great question. Errors in billing codes happen all the time. When we looked at patients who we knew had type 2 diabetes, many of them would have a spurious type 1 diabetes code or vice versa. And that's one of the more common types of errors you see. There's also things when someone explores a diagnosis and then doesn't end up getting that, but gets something else. And so one of the things that becomes actually really helpful with a simple metric to define a disease like ICD codes is requiring multiple codes on different days. So when you get billed for something more than once, especially on different days, it raises the probability of that being correct quite significantly. In addition, there are certain conditions that you could see that often do kind of collide, like type 1 diabetes and type 2 diabetes. We would see this also with Crohn's disease and ulcerative colitis as they kind of worked out the diagnosis in an individual's history. And there, when you have those kind of what I'll call sort of colliding or frequently co-occurring conditions in someone's record, you often see that the number, if you just do a count of the most common code amongst these pairs of codes, you often get towards the right diagnosis too. And sometimes that's just working out the diagnosis in time in an individual's life. And sometimes that's also just the number of errors that might happen over the true positives that get recorded of the correct diagnosis. And then the final thing I'll say is statistically. So statistically, when you have a incorrect diagnosis, you're usually going to bias towards what we would call a null result. So not finding the signal, which is the kind of error you would like to have when you have a billing error in a test of an association. So you're just going to be overly conservative in the signals you find typically.
Thank you. That makes sense. What are the variety of inputs that one can use?
People have done PheWASs with all sorts of things. People have looked at lab results, and that can even give you a sense of, for instance, we know elevated white counts are associated with all sorts of things. But, when you get to certain ranges of white counts, people have shown this, it's more associated with different kinds of conditions than others. Those really high white counts are going to be more things like your C. diff colitis than your common cold, as a very broad example, right? And people have looked at conditions. So look at one condition associated with another condition in a phenome-wide manner. People have looked at the polygenic risk scores. When you take a collection of genetic variants, which is often done now to report to a given disease, it's interesting to see how that looks across the phenome, because what you end up recovering are all the different kinds of associations against that given phenotype.
And I'll give you another example that I really think is a pretty one. And that is, as you may know, the human phenotype that has had the biggest genome-wide association studies to date is height. So height is a highly heritable trait. We all know that. If your parents are tall, you're likely to be tall. But it turns out there are hundreds of thousands of variants that each contribute a tiny little bit to how tall you are or not. And investigator Jonathan Mosley created a polygenic risk score for height. So he asked the question, can I create a combination of hundreds of thousands of variants that together predict your genetically predicted height? And then he applied that across the phenome. And one of the strongest signals was, to his surprise and to our surprise, atrial fibrillation. And it turns out that there's a well-established literature that tall people are much more susceptible to atrial fibrillation than short people. So that's known. And so it's an interesting way of getting at the same kind of information. But you would not have anticipated that. But that's an example of something that comes out of that. Now, what the biology is a really interesting question. And that's the kind of observation that a PheWAS can enable. It can enable somebody to sort of think hard about what it is about the genes that determine height that also determine susceptibility to atrial fibrillation. Maybe it's something as simple as how the heart hangs in the chest, or maybe it's something more complicated.
Thank you both. I think that's very illustrative. What are some of the clinical applications of PheWAS? We're thinking about all these examples. How are clinicians using this or going to be using this? What do you anticipate?
One clinical example is thinking about drug repurposing or thinking about potential drug side effects. So if you know a certain drug targets a certain gene, then you can look at genetic variants in that gene and see what diseases are associated. And you can look at both sides of it. It's not just what it potentially could treat, it's also what the side effect of the drug might be. And so, pharmaceutical companies are using PheWAS in large data sets to help them think about potentially de-risking a given drug target of potential side effects, and also thinking about what the indications might be. That's one example.
Yeah, I don't think that we're going to be doing routine PheWAS as part of clinical practice, just like we don't do routine GWAS. These are tools that build on clinical databases. The downstream spin-offs may be very, very important for the way we practice medicine, in the sense that we'll understand relationships among diseases, we'll hopefully be able to develop better risk predictors for individual diseases or better drugs to target specific disease mechanisms. So I think that's the hope of genome science.
I think that's a great point about the drug development and how this might be used or not used in clinical care. How do you hope otherwise that PheWAS will be developed and applied in the future?
I think one of the real opportunities is making it more routine and making these catalogs more available. When we started PheWAS, there were very few electronic health records linked to genetic data, and electronic health records have now become a almost commonplace part of large biobanks. And so that means there are just much, much larger data sets that are becoming available with genetic data and health data as routine and also more accessible. These are, of course, powered by lots of participants who are saying yes to being in parts of these kinds of studies. But these resources are starting to become available in large ways. And the most available to date is something called the UK Biobank that has recruited 500,000 people across the United Kingdom. and they have genetic data, health data from their healthcare system, and lots of survey data, other kinds of things. In this country, we've launched the All of Us research program, which has over 500,000 participants, and we've released our first genetic data set, which has 100,000 people that have whole genome sequencing in it. And what's really notable about this set is that, half the people in the data set identify as race or ethnicities other than white. And most all genetic studies, 96% in fact, are driven by individuals of European ancestry worldwide. So this is a real chance to sort of expand things like PheWAS to diverse populations and to understand the diverse genetic architectures around disease in a more complete way. And that will lead to better understanding of genetics, better understanding of potential treatments, and just have a wealth of effects from it. And so I think PheWAS is coming back to that is just one of those tools that accelerates our understanding of genetic architectures. It'll be especially important to see this applied, I think, over more diverse ancestries, which is something that's been very much missing in genetic studies so far.
You bring up very important points, very clinically relevant. Dr. Roden and Dr. Denny, thank you again for joining us today to discuss this interesting topic. And thank you to all our listeners. I want to remind the listeners that further details may be found in the article by Drs. Roden and Denny and Lisa Bastarash, titled Phenome-Wide Association Studies in the January 4th, 2022 issue of JAMA. This episode was produced by Daniel Morrow at the JAMA Network. The audio team here also includes Jesse McQuarters, Shelley Steffens, Lisa Harden, Audrey Foreman, and Mary Lynn Ferkaluk. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com. Thanks for listening.